protein structure is more complex in some ways than the other three macromolecules. When we talked about carbohydrates, remember the polymer, the polysaccharide, was really just a simple chain of the monosaccharides, and there wasn't much more complexity than that. Protein structure is more complex because proteins have a very specific three-dimensional shape. And if that three-dimensional shape changes, then that protein doesn't function correctly. Shape is key in all of biology. The shape of a molecule is what the cell recognizes. So if that protein isn't shaped correctly, it's not going to function the same. So let's look at 4 levels of protein structure. The first level of protein structure is called the primary structure of a protein. And the primary structure of a protein is really just what is the amino acid sequence of that protein. Okay, so looking at this polypeptide, it's a chain of amino acids. The primary structure is saying which one goes first, which one's second, which one's third, fourth, fifth, and so on, all the way down this chain. So that polypeptide chain has a specific amino acid sequence. And the primary structure is simply what is the amino acid sequence of that protein. If even one amino acid in that chain is different, that protein is going to fold differently, have a different three-dimensional shape, and not be the same protein. That amino acid sequence has to be correct. It has to be the same in order for that to be the same protein. And that is called the primary structure of a protein. The secondary structure of a protein is next. And the secondary structure is a little more complex. In the secondary structure, there are some regions of proteins that coil due to this specific pattern of hydrogen bonding that occurs. And there are some that actually fold into a pleated sheet. Those are called alpha helix and beta pleated sheet. And when a protein has one of those two patterns, or both, that is called the secondary structure of that protein. So secondary structure is going to be an alpha helix and the beta pleated sheet. And both of these involve hydrogen bonding. Remember when we talked about hydrogen bonds, I said we're going to see 3 major molecules at least in which hydrogen bonding occurs: water, proteins, and DNA. So here you go. Hydrogen bonding in a protein is creating that secondary structure. The third level, is called tertiary. The tertiary structure is the specific three-dimensional shape of that protein. So the three-dimensional shape of that protein is called its tertiary structure. It's also called the conformation of that protein. Conformation is another way of saying three-dimensional shape. That specific three-dimensional shape is the result of the amino acid sequence, but more specifically, the reactions between the R groups of those amino acids. So if you can imagine, we have this chain of amino acids that produce the polypeptide, and they each have an R group. And when those all get in a line, those R groups are going to start reacting with each other. I always like to think of preschoolers standing in line waiting to go on a field trip or waiting to come back into class. And you get them all in a line, and there's no way for them to not start reacting with each other. They're going to start tickling each other and poking each other and punching each other, and before you know it, you have this heap of preschoolers. That's kind of what happens with the protein. These R groups are all going to start reacting with each other in a very specific manner based on their chemical properties. Remember, we had some that were polar, some that were nonpolar. Well, obviously this protein is going to be in contact with water. So all of the hydrophobic amino acids are going to tend to fold on the inside, away from the water, while the hydrophilic ones, the polar ones, are going to be on the outside. Different bonds are going to occur between the amino acids. So maybe this one might react with this one and it will fold around that way. This is all ultimately going to result in the three-dimensional shape of the protein. And then finally we have something called quaternary structure. And I'm going to write this in red because not all proteins have quaternary structure. Quaternary means fourth. This only occurs in some proteins. All proteins have 1 through 3. Okay, all proteins have a specific three-dimensional shape. All proteins have a secondary structure. They either have alpha helix or don't, have beta helix or all proteins have these first three levels of structure that should be considered. But only certain proteins have quaternary structure. Only those that consist of more than one polypeptide chain. So quaternary structure is only in proteins with more than one polypeptide chain. Most proteins are just one polypeptide chain folded into its specific three-dimensional shape. But some have more than one. Good examples are collagen and hemoglobin. Collagen is actually 3 separate polypeptide chains braided together. And hemoglobin is 4 polypeptide chains. If you have more than one, we would call that the quaternary structure of a protein. This just shows you an example of the different amino acids reacting with each other to produce that three-dimensional shape. So the R groups reacting with each other. In this case, a disulfide bridge is forming. Here an ionic bond is forming. The hydrophobic ones are tucking inside. A hydrogen bond is forming there, so that would be probably part of the secondary structure. This is collagen. This shows the 3 separate polypeptide chains, the blue, the green, and the red, that make up that protein. So collagen would have quaternary structure. This is showing the 4 polypeptides that come together to produce hemoglobin. So hemoglobin would have quaternary structure. This is a very, very important concept. I mentioned earlier that if even one amino acid in this primary structure is different, that protein is not going to have the same shape. And you can see in this example, amino acid number 6 is different. And just having that one amino acid out of thousands be different results in a different shape of that protein. In this case, that protein is hemoglobin. And you can see normal hemoglobin versus sickled hemoglobin. So this is sickle cell disease. In sickle cell disease, there is one amino acid that is wrong due to a base on the DNA being different. A different base on the DNA gets transcribed into a different base sequence on the messenger RNA, And then that gets translated into a different amino acid in that polypeptide chain. And just that one difference results in a sickled red blood cell that does not carry oxygen properly. So one amino acid being wrong can make the difference between having a disease and not having a disease. Having a normal protein structure, not having a normal protein structure. So that amino acid sequence is very, very critical ultimately in the three-dimensional shape and the function of that protein. Okay, so this is a normal protein, and when we talked about pH back in the properties of water lecture, we talked about the fact that pH levels need to stay constant in different regions of the body, otherwise protein structure is affected. When we have changes in certain conditions in the body, the protein structure can change. If you think about cracking an egg white into a frying pan and seeing how that egg white changes due to that increase in temperature, that's a very good example of what we're talking about. That process is called denaturation. When a protein becomes denatured, it means its three-dimensional shape changes and it might no longer function properly. In fact, it probably won't function properly. Denaturation is going to be a change in the tertiary structure, which remember is the three-dimensional shape of a protein. This is usually due to changes in temperature. So due to changes in temperature, pH, or salinity. Salinity meaning saltiness, solute concentration. So temperature needs to stay constant in the body. pH needs to stay relatively constant in the body. Salinity needs to stay relatively constant in the body. If not, we denature that protein. Denatured proteins do not function correctly. And if you're talking about an enzyme that becomes denatured, that chemical reaction is not going to take place. If that enzyme can't bind the reactant, then that chemical reaction is not going to take place. So this is a normal protein, and then this would be a denatured protein. You can think of it as unraveling. That three-dimensional shape changes and that protein no longer functions. So proteins function in a very narrow range of temperature, pH, and salinity. It's very important for the body to keep these factors constant for the proteins to function correctly. It's especially important when we're talking about enzymes. So let's look now at the connection between DNA, RNA, and proteins. So in the nucleus of our cells, we have DNA. So I'm just going to draw a rough cartoon sketch of a cell here. So plasma membrane, nuclear envelope, and now here's our DNA. DNA is a nucleic acid. DNA is in the nucleus. It never leaves the nucleus. The DNA is the master recipe for how to make every single protein in the body. That is what our DNA is. DNA codes for proteins. This is our only copy in each cell for how to correctly make that protein. And you already saw one example of if one amino acid is wrong, that protein is not going to have the same three-dimensional shape and it's not going to function properly. So shape is key. We have to have the correct amino acid sequence. That amino acid sequence is coded for on the DNA. So what happens is we have one region of DNA called a gene. And that gene is read every time we need to make a protein. But we don't want to send this DNA out here in the cytoplasm to put that protein together. So we have all these amino acids out here we got from our food or from recycling other cell parts. We have all these amino acids out here, and we want to put this protein together. But we don't want to send the recipe for how to make it out here and have it get damaged. That's the only recipe we have. So what we're going to do instead is we're going to send a messenger out. And that messenger is called messenger RNA. So what happens is here in the nucleus, we're going to make a copy of that recipe, and we're going to send that copy out to the cytoplasm. And that copy is called messenger RNA, which is also a nucleic acid. That messenger RNA is going to come out into the cytoplasm. So you can see here, we made a piece of messenger RNA, and that's now going to come out to the cytoplasm of the cell. And a complex called a ribosome is going to assemble around that messenger RNA. And it's going to read that recipe. And it's going to start assembling that polypeptide based on the base sequence that occurs on this messenger RNA. This messenger RNA is saying, put this amino acid here, now put this one, now put this one, now put this one. That messenger RNA is determining the amino acid sequence of that polypeptide. So out here in the cytoplasm, this messenger RNA is going to be read by a ribosome. We have millions of ribosomes out here in the cell. And as that feeds through, it gets read. And it's telling it, put this first, this second, this third, is determining the amino acid sequence of this growing polypeptide. Okay, so here's our polypeptide. And then obviously that will ultimately fold into the three-dimensional shape of a protein. Once it's all done, all those R groups start reacting, and we end up with the three-dimensional shape of that protein. So this is how DNA, RNA, and proteins are connected. A couple of important terms you should go ahead and learn now. DNA becoming messenger RNA. That process is called transcription. And transcription takes place in the nucleus of the cell. Now that messenger RNA comes out to the cytoplasm, and the ribosome complex assembles around the messenger RNA, and messenger RNA becoming a protein is called translation. That translation takes place in the cytoplasm on a ribosome. So this is how DNA, RNA, and protein are connected.